And I would especially like to thank George and Esper for organizing this great meeting. I always want to say too much in my time period, and I think today I'm really over the limit, so I will do my best. I cannot speak Portuguese, but I can do Google Translate. So my slides will be in English and Portuguese, so that my colleagues from around the world can understand what I'm saying. So one of the things that we've been doing is looking at imaging at multiple scales. Oh, it's not working right. I think it's too big. Anyway, it'll load up. This is a half of a gig presentation. So we can go from whole animal models, a mouse, to macaque tissues, and I'll be talking about this, down to cells in tissues and in a culture dish. And recently I'm like a child that got not the best toy at Christmas, but the two best toys at Christmas. Woohoo! And so now I can do PET scans and can do light sheet microscopy, multiphoton imaging, and we can go from a whole animal down to we found a foci of infection down to the viruses at the EM scale. So we developed a system to find early foci of infection using a vector. And so it's luciferase, which allows us to find the site of infection. And then cherry allows us to find the individual cells that have been transduced by this vector. And so this is a new way to look at transmission. And so these are transductions. This cannot replicate. So any events you detect come from the inoculum of the challenge. That has advantages and disadvantages. It is a high titer stock. We can do vaginal and rectal challenges, and the studies are done in collaboration with Ron Veazey at the Tulane National Primate Research Center. They send the tissue to us in Chicago to begin the evaluation. So this is an example of the data. So here is one half of a female reproductive tract. And this is a biopsy site. Then we add the luciferin, and we can see photons being generated. This biopsy is generating a lot of photons. And then we can take this piece of tissue here, cut it into smaller and smaller pieces down to 2 by 2 millimeters. And then the 2 by 2 millimeters, it's very easy to go through that tissue completely and find the transduced cells. And so then we can go into the tissue, and we can show that the cherry fluorescence is being expressed. We can validate that by spectral imaging. We can stain for luciferase to show it should have both proteins, and then we can phenotype these cells. And what we discovered is that essentially this vector is mapping places in the epithelium where the virus can get through. So we can mix SIV MAC239 wild-type virus with the vector. And so you can see here that when we get a PCR signal for the vector, we can find lots of the real virus, and then we can go in and stain for the virus and find the infected cells. So this is, again, we want to validate all of these signals we're detecting. And so in this case, here we have a cell that has been infected by SIV. It is expressing envelope and GAG. And then green is CD4. And so one of the things that happens as a consequence of infection with HIV or SIV is downregulation of CD4. So you can actually see that in this cell, instead of being on the surface, the CD4 has been internalized and then it gets degraded. We can quantify that relative to the CD4 cells that are not infected with SIV. We see a shift down in fluorescence, and then this radius of gyration, the area of the GFP or the green signal of the CD4 is decreased. So this validates this is behaving like an infected cell in vivo. So we really are finding infected cells. So we then went on to try to phenotype the cells that are being infected. and in all of these different tissues. And really what we find is that Th17 cells are the primary target. So even though they only represent 18% of the T cells present in these animals, they represent 84% of the T cells that are being infected. So there's a five-fold enrichment with the SIV. Why Th17s? And we also see immature dendritic cells. And we have a lot of work to do, and this has been discussed quite a bit at this meeting, but these are metabolically active cells. These cells are involved with epithelial barrier homeostasis and maintenance. And so the Th17s, basically the immature dendritic cells are capturing commensal bacteria and other antigens and showing them to the Th17s. If they see too little antigen, they say to the epithelium, stop, you don't want to get too thick. If the epithelium gets too thick, you can have plaque psoriasis. So underneath every plaque of plaque psoriasis is a bunch of Th17 cells that are working too hard and making the epithelium too thick. So these cells are doing a job, and so they are active, they are awake, and these are good targets for the virus. And these cells are known to be the first cells depleted in SIV and HIV infection. And if you isolate the cells out, they will be also infected. And you can infect them efficiently with a VSV-G pseudotype virus. So what that says is that what's important for the infection of these cells is not what's on the surface, but what's on the inside, the metabolic state. Nucleotides reverse transcribe, import pathways to get it in. It allows the virus to go through its cycle. So we have recently extended this work to the rectal compartment, and the key was to use a fluorescent protein that was further red, a near infrared, because fecal matter, poo, is fluorescent. So you have to avoid that signal, and so we can do that. It's the same sort of a thing. We find transduced cells both in the near the rectum and anus, which is a squamous epithelium, and then in the rectum itself within these crypts. And we can then phenotype these cells for the different markers. And in the end, what we see is, again, for rectal challenge, Th17s are the primary targets, and CD4s are the major targets. Most of them are Th17s. Some others, and this is very similar to what we have seen in the female reproductive tract. So there's this consistency of the target cells. These are viruses that infect through mucosal sites, and they selectively infect first these cells that are really only present in high concentrations at mucosal sites. So in the rectal studies using the vector, what we can see is this five-fold enrichment of Th17s, and then there's fewer immature dendritic cells present, but we see a similar five-fold enrichment. So these two cell types are the preferred early to be infected early. All the other T cells, they really aren't being transduced well at all. So this is really the preferred target. So this just gives you a sense. This is now looking at later time points. So everything I've shown you was 48 hours after challenge. And now to get a sense of the dynamics that are taking place, we are looking at 48, 72, and 96 hours. 2 days, 3 days, and 4 days. And this, by 4 days, it just becomes green. Here are SIV-infected cells. The red are T cells, both CD4s and CD8s. And you can see this is a battlefield. Like here, and this is what, you should look at some of these cells, Tim. This cell is undergoing apoptosis. The cell is exploding. There's all these fragments around. You have to really be careful as to what you interpret as an infected cell, or was an infected cell, or could be an infected cell. There's a lot happening. And to demonstrate that, if we look at comparing 48, 72, and 96, the Th17s are kind of preferred as long as they're around. we see for the immature dendritic cells, they are no longer really being infected at 4 days. And this is very consistent with some of the work with Ashley Haas, for instance, where the earliest time points he published was at 4 days. And he saw almost all T cells being infected. And then these other non-Th17 cells begin to wake up or migrate in. And so they start to become the primary targets at this later time point. So the dynamics here is quite great in the first several days after infection. So why are some of these cell types preferred? Well, most of the effector memory cells are sleeping. They're in a resting state. They are hoping to someday bump into their antigen presented in MHC and to initiate an immune response. But these other cells, the dendritic cells, the Th17s, they're working all the time. But it looks like by day 4, these other cells are starting to wake up. We can study the dynamics here, so we can see at 48 hours, so this is really early, we can see infected cells, we can see apoptotic fragments, we can see phagocytosed cells, and then we see these lyse cells. This is another one that will look similar to some of what we saw yesterday. These are just cells that seem to have exploded. It could be pyroptosis, and so now we'll rotate this, and you can just see it's like a disc, almost looks like a supernova, a disc of GAG and envelope that this cell has exploded and gotten crushed between the adjacent cells. So we can quantify these different species. This is a very nice example of a phagocytosed infected cell. So here this is a immature dendritic cell, CD11C positive, but it contains a SIV antigens associated with CD3. So this is a CD3 cell, or an infected CD4 cell that has been eaten by this dendritic cell. Here are some of those supernovas. And so at 48 hours post-infection, we can see this sort of a spread of the different components. And the problem is we don't know the half-life of these, how long it takes for the body to clean up the antigens. So if it's fast, some of these populations could be more abundant than they seem because we're just taking a snapshot. So the other thing that's becoming very clear, so normally if you look in the mucosa, there's very few NK cells, for instance. But if you look at 4 days post-infection in these foci where there's this war going on, all of a sudden now you have abundant NK cells showing up, and you also have abundant recruitment of neutrophils. So typically in the rectal compartment and the vaginal compartment, there's not a lot of these cells around, but when you have this foci of infection, they come migrating in, and I suspect there's T cells are also migrating in, and it's a little bit like the first responders at a big fire or something. They're sort of going in, and at least some of these cells are not going to be coming out. So we can use this reporter system to identify susceptible cells. And we find that there's Th17s and other metabolically active cells. So now we have a nice system to study transmission and early events. And so now we want to understand several things. One of the things is host responses, what's happening with the host, but also really want to understand the mechanisms of prevention, topical prep, microbicides, oral prep, systemic prep, antibodies, vaccines. We can now start to see how they perturb these early events that could lead to prevention. So the one example I want to talk about is an intravaginal ring that was developed by my friend and colleague Patrick Kaiser. It has TDF, and so the vaginal ring is a form of microbicide delivery. The idea is you deliver a high concentration of the drug locally, and then those cells should be protected. In a macaque model, these things work perfectly. In reality, they work very poorly. And there's been a lot of suggestion that these things don't function because of improper use, but there's a lot of biology emerging. The microbiome can eat the drug, metabolize the drug, and so it's gone. You can have inflammation that can overcome the possibility of drug. There's going to be a paper in the April Nature Medicine, and I have a news and views there describing some of these biological bases for why microbicides fail. So what we're going to do is a comparison of oral prep to topical prep, and we're going to use Viriad, TDF. Okay, and so this is unfortunately because of budget limitations and access to pigtail macaques. This is a smaller study, but it very much recapitulates some of our other work. So we're going to have 2 control animals, 3 that got the ring, and 3 that took oral TDF, and then we're going to look 72 hours after vaginal challenge to see what are the infected cells, where are the infected cells, and how does systemic PrEP differ from oral PrEP, or is there any difference? So this just shows the oral PrEP was TDF, and it was 22 milligrams per kilogram, and this showed a good protection in one study. And these are the data from the animal. So they put the drug in a treat for the animal that they like to eat. So you make sure they're going to eat the drug every day. And this just shows that we had good drug levels in the vaginal fluid and tissues and cells. So what we can do here then is basically in vivo PK/PD. We can ask in these tissues, where is the drug and how much is there? So these dots, this scale, is for the amount of drug present. And so these 3 are from the ring. And so you can almost see where the ring was lying, right? So the ring is here, just below the cervix, and that's where the highest concentration of drug is. Unfortunately, very little drug makes it up into the uterus and to the ovaries. This is now systemic PrEP, and you can see that there is no very high signals, but sort of generally similar signals throughout the tissue. So this nicely illustrates this difference. And then what we can do, and this is ongoing, is we can go into these tissues, we can find the sites of infection, we can do PCR to kind of map everything, and so we can get a sense of, again, where is the virus, how efficiently is replicating in the influence of these treatments. So this just shows a control animal at 72 hours. We have big foci. These red dots are the SIV infected cells, and we have lots of infected cells, very big foci, as we have in some of our other studies in rhesus macaques. And we can just go in and phenotype them. This is now an animal that has the ring, and we see very little infection in the vaginal vault where the drug concentrations are high. But we see this is a piece of uterine tissue. All these red spots validated by spectral imaging is a very large foci of replication of SIV MAC 239 72 hours after challenge, vaginal challenge. And again, we can phenotype these. Now, when we start to look at oral, we see a very different picture. We have to look harder to find infected cells, even though it's sort of telling us where to look. And we just find, here's 2 infected cells, but no big foci. Again, just 1 or 2 cells here, very small foci. In a couple of places, we'll find more cells. Very often, it's right at the lumen, or in a draining lymph node, we can find more cells. So the preliminary conclusions are that large foci of infected cells are seen in the control animals throughout the female reproductive tract, and further away from the ring, such as in the uterus or the ovaries. In contrast, for systemic PrEP, oral PrEP, we see very small foci whenever we find them. So what is possibly happening here? So I think this is the peril of having a large this large local concentration of drug, because if the virus can get through, then there's nothing to stop it. So it's a little bit like you have a wall. If there's a weak point in the wall, it's too late. You can't stop the invading horde, right? But when you have oral PrEP, you have a different sort of a circumstance. And I think a nice way to think about this is when you are giving oral PrEP and you want to say get IC90, IC95. And what that means is 95% of the cells are protected, or 90% of the cells are protected. And that's the part everybody likes to say. But if 90% of the cells are protected, that means 10% of the cells are not protected, okay? So, what we believe we're seeing, and the work is ongoing, is that those few cells we're seeing are the cells that don't have enough drug. to prevent infection and replication. And but as long as there's below a certain threshold of cells to infect, the virus can't spread. The virus can't go systemic. The virus can't take over the body. And so I think a nice analogy for this is the herd effect in vaccines. To protect the community with a vaccine, you don't have to vaccinate everyone. You just have to vaccinate most people. So if you have 2 infected people and all these blue individuals don't have the vaccine, all of a sudden everyone's infected. If you have a few with the vaccine, you can't stop it. But when you have most people vaccinated, the virus can't go from this one to the unprotected person. So this is how I think we need to think about this. So this is another biological method or a biological explanation for why microbicides really never achieve protection, whereas with oral PrEP, it's very clear, the epidemiology, there can be greater than 90% protection, 97% protection in some cohorts. So now I want to shift to protection from antibodies and some methods we've been trying to develop to follow antibodies, and we now have this working really very well. I want to give a shout out to John Mascola and the team at the VRC, who has been exceptionally generous with sharing antibodies, VRC01 and VRC01-LS, the antibody that persists longer. And if we were to buy these antibodies at the corner store, we would not be doing these experiments. So this really has been very important. And so what we're doing is just putting a floor onto the antibody, like you would use for flow cytometry or immunofluorescence. You just put one floor on the antibody, and then we can inject it into the animal and watch over time. And so can you guess which animal got the SIV labeled antibody? And in white light, I'll tell you SIV is blue. So it's very easy to say, I pick this one. This one got the antibody, and this was the control. And we can draw samples at different time points and put it in a fluorometer, and here you can see the fluorometer, and you can see the different colors, and it goes down over time. We published a paper late last year to show that really the floor on the antibody does not, in a large way, perturb the function of the antibody. For VRC01, I think there are 32 lysines, primary amines, that can be labeled. So, and the idea is it randomly goes here, here, here. So even if one of those amino acids is very important, you're only getting rid of 1/32 of the, I think about 6% of the antibody. And then one of the things that we've learned very quickly, and this came up a little bit yesterday in Rick's talk, is you really should wait 1 week for the antibody to become systemically distributed. And I think a lot of the curious results that people are trying to understand out there are because the antibody is catching up to the virus. So you see, Dan Baruch published a paper in summer of 2016, which they injected the antibody and challenged 1 day later, and they showed virus spread that stopped after 1 week. My hypothesis is that the antibody finally catches up to that virus and can stop it. And I think everybody, whether it's systemic PrEP or these antibodies, everyone would like it to be sterilizing, that we see no infection at all. I don't think, even think about how the immune system works. Sterilizing immunity probably isn't a real thing. It's not even possible. So, but the good news is that the antibody can catch up to the virus. The drug can catch up to the virus or prevent it from going systemic. So, sort of the beginnings of this was trying to understand antibodies in the vaginal compartment. And this was before John started to share those wonderful monoclonals. So in these studies, we used bulk human IgG, Gamunex. And Gamunex is used in a variety of therapies in humans, individuals that don't make IgGs. Also, if people have autoimmune diseases that are antibody-mediated, they can take Gamunex and increase temporarily their antibody levels by 3 or 4 times of normal. And in days, and definitely in a week, it'll go right back down to normal, and we'll touch upon this in a second. But it goes back down to normal, and you get rid of 3/4 of the bad antibodies, and so you'll have less symptoms from the antibodies. So we knew that there was antibody concentrating here near the lumen, and we knew it was down here in the lamina propria under the tissue, but was it coming from the top down into this, or was it coming from the bottom up? And as you looked at these images, it really started to seem like it might be coming from the bottom up, which was unexpected. I love these imaging experiments because you just look at these things and you're like, what? And then you slowly work through it, and then we can like learn new things. So it's one of my favorite things to just look at these things and just try to understand what's going on. So here you can see we have biopsies from the same animal, 24, 48, 72 hours, and 1 week. And so we're now gonna, this white line is where the lumen is. This is the vaginal vault. We're going to amplify the E-coherence signal because this is where the granulosum is between the stratum corneum. So we're going to draw another green line, and then we're going to increase the signal for the antibody. And so you can see there's a little bit of antibody above this zone at 24, more at 48, more at 72, and by 1 week it's completely throughout the stratum corneum. So the antibody that's there is coming from the bottom up. And then if we look closely at some of these images, it seems to be coming in a very unexpected mechanism. And so you can see here again the antibody. These are clearly cells that are dying by apoptosis as they're supposed to, these remnants. And then you can see that some cells here in the basal cells at the lamina propria are gathering a large amount of antibody. And then we believe that they then migrate up here and die. And so they're delivering the antibody there. So if we get rid of the other signal, you can see that in some places you have these cells that they migrate up, and these are cells that are undergoing apoptosis, and they deliver their contents here that as the cells differentiate, this sloughs off, it goes up, it delivers antibodies ultimately into the vaginal lumen. And I'll show you some fascinating observations in the macaque model in a second. So you can say, well, okay, so this is antibodies you're injecting, and the neat thing is, it's like, okay, so it's like now you inject the antibody, and you can watch where does it go in 5 minutes, 10 minutes, 1 hour, but it's not really a normal system. But we can just go into these tissues and stain for IgG antibodies, and we can find that this is happening in our wet squamous epithelium all the time. So in human vagina, we can find these cells containing antibody. In macaque mouth, which is wet epithelium, squamous epithelium, we can find these cells. In the human inner foreskin, we can find these cells. And it was fun. We found a paper from 1979 that was describing this serum proteins in squamous epithelium that they didn't really understand what it was. But it's nice to know that other people have seen this. Even that was over 30 years ago, almost 40 years ago. So this also appears to be regulated because sometimes we see these walls of antibody. And so you can just see that many of these cells grabbed antibody and migrated to the surface. You can, when we first saw this, we were thinking that it was as a consequence of some disruption of the epithelium. But in a minute, I'll show you with one of the new methods that it might be more complex than that. So one of the two new methods is something called light sheet microscopy. And so essentially you can look at a bigger piece of tissue, centimeter-sized pieces of tissue. And so, and to do this, you need to clarify the tissue, and you extract it with different solvents. And then we can take these tissues and put it on a Odyssey detector that sometimes people use for westerns and other methods, and you can see that the patterns of the SIV signal, the antibody in the tissues, is not spread throughout. They have these distinct patterns. So here's an example. So you just kind of scan through the tissue, and so this is rectal tissue. Green is the antibody. One of the things I love is you can see the antibody showing up in the mucus with this. Watch how it's out in front of the cells. This is antibody showing up in the mucus, and it is concentrating, we believe, and we have other data showing in goblet cells. And these movies are awesome. It's just like so much fun. And you just stare at these things, and I'll even do that one again, even though I'm going to waste time. But you can just see so many details, and we have been trying for many years to see antibodies in mucus, but mucus is very hard to fix, and it turns out the methods of the clearing tissue gives us the mucus signal. So now what we're doing is looking at different time points to get a sense of the distribution and looking for differences between VRC01 and VRC01 LS. So we can either do one at a time, or we can label VRC01 one color and VRC01 LS another color, and then watch them going together. So now this is looking at different time points, and this is at 24 hours, and you can see these strange flashes. I call it tissue lightning. So you can see this by 72 hours, it's really quite different, and you can see some of these flashes, but there's more antibody is now in the tissue compared to here over time. So now we can do a three-dimensional reconstruction of this, so now we can look at how everything lays next to each other. And so this is an example of 24 hours, and what it reveals, those flashes, is just the Z series through the pipes that are delivering the antibody into the mucosa. And then by 72 hours, we don't see the piping so clearly, but we see this, I call it feathering, this strange concentration of signal here at the tip. And again, more antibody is delivered by 72 hours compared to here. You know, we're learning these things. It's a lot of fun, and so you go look in textbooks. And I guess textbooks are oversimplifications because, you know, the textbooks show one of, you know, if this was vasculature, you would see a artery going up, and then a capillary, and then a venule coming down, and it should be wrapped around each crypt. So that's why at first we thought this might be draining lymphatics or something more exotic. You know, I was talking about reverse osmotic flow of the draining lymphatics. And it actually turns out that this is really the normal circulation. And I'll hopefully convince you of that in a second. So first, the feathering. And here's an example of that. It's very close to the surface. And what we think we're looking at, this feathering, is actually cells from the blood that are doing the classic high endothelial venule bind, roll, and crawl through the endothelial wall. So this sort of a process that happens in the high endothelial venules. It is, you know, in all this vasculature here, you can see that some of it is high endothelial venules. We're doing the experiments right now, but we suspect that what we're seeing here, and when you look into higher mag, that this is high endothelial venules, and it just takes a while for the signals to accumulate, and these things move through slowly. Now, for the piping that we saw, the key came from looking at paraffin sections. There's a lot of reasons why I hate paraffin sections, but now from some of this new data, it's become a critical tool, and now I'm forced to change my mind and love paraffin sections. And the reason is that when we do our normal experiments, we are washing away the contents of these tubes. But in a paraffin section where everything is fixed and not going anywhere, we can see things like the red blood cells. So you see red blood cells in the tube. Here is red blood cells that are in a capillary, and capillaries are, it took us a while to figure out what we were staring at here were capillaries. And then one of the fascinating things about the capillaries is there is a concentration of the LS antibody in these capillaries relative to VRC01. So, and this is probably more of a high endothelial venule. These aren't bubbles. These are probably lymphoid cells that are starting to attach to the inside of this vessel, and they exclude the fluid that contains the antibodies. So very likely, so LS interacts better with FcRn, and FcRn has been suggested to do a bunch of things, but I think the one thing that it very clearly does is maintains IgG homeostasis. So when macropinocytosis of fluids in the blood is taken up, it is internalized, and then as this pH becomes acidic, FcRn will bind to antibodies or serum albumins and then recycle them back into the fluid. Whereas the antibodies that aren't bound to FcRn and other materials go to the lysosome to be degraded. And so we wondered if we might be able to see this segregation in some of our tissues. So in this experiment, VRCO1 is going to be green, and VRCO1-LS is going to be red. And so the idea is they will come in together and be yellow, and then would be through recycling endosomes, the LS would be salvaged and sent back out, or importantly, transferred into the tissue on the other side. Just like FcRn takes antibodies from mother's milk in the gut, grabs the antibody, brings it in, and doesn't put it back out, it sends it to the other side into the tissue. So that's how one of the ways that the baby gets the mother's antibodies. The same thing happens in the placenta. So that's, I think, an important, another important piece of information. So can we see this segregation within the tissues? To do this, we used a brain tissue. And so this is a capillary and brain tissue. And so what we can see here is structures that look like the recycling endosome. And you can see places that are yellow where they're both together, and you can see them being separated here. And then you can see lots of areas where there's red, where this is being sent either back into the blood or out into the tissue. Very little green alone, very little red and green, because that is being degraded in these cells in this system. So I showed you this picture earlier, but we see accumulation of LS within these capillaries, and we can, when we do these mixed experiments, we'll do LS red, VRCO1 far red, and then we'll switch them so we can go to the different tissues and be sure it's not some strange artifact of the fluorescence or something. And so these capillaries are where we're seeing this accumulation. And then if you go back to the first papers about FcRn, they specifically say it's expressed in capillaries. And that makes sense because it's not, the blood flow isn't so great, it's this macropinocytosis and the gathering of the fluids is much easier in the stiller waters of a capillary than in a vein or an artery where the blood is just flying by at very, very high rates. So, and then to prove this, and we're doing this on all the tissues, but essentially this is a marker for capillaries and other vessels, and so we can see that these are in fact capillaries in the brain. And the reason we like to use the brain is that antibodies are getting there, but they're getting there at a slower rate. So we can show that these structures that we're seeing are capillaries, and they're very strange visually because they're not thick like a artery or a vein or arterial or a venule. They're just these very wispy spiderweb looking kind of cellular structures. So now this is a light sheet image of a piece of vaginal tissue from a macaque, and this is from 3 days after injection. And so we actually, a whole lab meeting, somebody put this up, the first light sheet imaging we did, and we just looked at this and I went, what is that? What are these things? And it was like, what's the top or the bottom of the tissue? And they're like, I'm not sure. So then I'm like, okay, let's go back to the microscope. I will come with you. Let's go look at it. And so it turns out that what we're seeing here are these, typically not during menstruation, in a rhesus macaque or other macaques, you have this very thick epithelium. And what we're looking at here is this feathering that is present. And so this is the exact piece of tissue. Not a very good, this is an iPhone picture, not very good. But you can see that these dots correspond to these tips. So we know what we're looking at, but still, what? But now you start to look at the light sheet, and what's really neat, the first thing is if you remember this little bit more 1 week, and so we can now look in the whole big piece of tissue, and so at 24 hours you see just this little bit of antibody signal right here. And then at 48 hours, you can see it's much bigger. And then by 72 hours, it's much thicker. And this is everywhere in the tissue. So sometimes you can say, well, Tom, you're showing me this little picture here, but it is everywhere. And then you can start to see something strange going on with these tips, these structures where the antibodies are concentrating in these peaks. So this is going to be another nice light sheet movie. And so you can Z through this, and you can see that there are these thick gatherings of antibody-containing cells that are concentrated in these structures as they're growing out, and then we can look at them more carefully, and you can see they are those cells like what we've been talking about. So in addition to being everywhere, there's these places where they grow up, and so those structures I showed you in the gross level in the tissue, I thought that was sort of what was left as the pieces of tissue flaked off, but in fact, these spikes are growing out of the epithelium. And so we can, oops, oh no. Yeah, you can just go right to there, it's perfect. All right. Perfect. So this is just some higher mag images with the light sheet is great because it shows you these structures, but it doesn't show you the details. But we can take those sections and put them in our regular microscope and start to see neat things happening. So this is, again, consistent with one of these spikes growing. You can see it. It's just areas where there's a lot more of those cells. So maybe some of those walls of antibody we thought we saw are actually these spikes forming. And then you can see at the tips of these spikes, there's just a whole bunch of antibody gathering. So I think this is one of the mechanisms of delivery of antibody into the vaginal vault. We've got some more work to do there, but and then for this, all of this, it's exactly the same. If we put LS and VRC01 together, they do everything the same way, which is different than the rectal compartment. All right, so now I'm going to talk about my other great toy, woohoo, that I have now, which is PET scans. So PET scan, it's working with a couple of different groups, but especially Francoise Villinger and Phil Santangelo at the New Iberia Research Center. This is one that was in the hospital. So it got refurbished, and now we can use it for monkeys. And so you have to put copper onto your antibody or whatever you want to study. You inject it, you scan the monkey, and so you can start to see where things go at the whole animal level. So this is some examples now. After 1 hour after this animal got LS, this animal got regular VRC01. And at 1 hour, you really can't see very much difference. But when you go to 48 hours, you now see lots of differences. So in the lung, in the vasculature of the lung, the LS is concentrating to high levels, and I suspect this is going to be similar to what we see in those rectal capillaries. That's a rich environment. There's lots of blood flow there. There's probably lots of those capillaries, and so that's why we're seeing this concentration relative to the VRC01 down here. The other place we're seeing increased localization is here in the gut. So we can quantify all this, and we're getting more examples right now, and we're doing some female animals to look at the female reproductive tract. But you can see that that we see differences in the accumulation of the antibody. So in certain tissues, like especially in the gut here, we see it increasing over time. We don't see that much increase with the regular antibody. So this is sort of all ongoing and some nice work. So the other thing we're doing is what we call correlative PET. So if you remember, we use luciferase to tell us where to look in the tissue, and then we go in and follow the fluorescence. So it just sort of tells us where to look. So now we're doing this with the PET and fluorescence, and so we can either, I just showed you, we can tag the antibody, or we can put copper on the virus and introduce it rectally or vaginally and watch where it goes. And before this, we would say, you do your experiment. It's like, well, look here, and these experiments that tell us that we are looking in the wrong places. And it's easy to find rare things. You lose your keys. If you know where to look, you can find them. But if you don't, you will spend forever and just get flustered. So this animal has gotten a rectal challenge with copper labeled, in this case, antibody. We see this very focal localization of the copper signal. We then cut this into little pieces, put it into cryomolds, and scan it. So we scan the whole animal, we take out the tissues, we scan the tissues, we cut those in little pieces, we scan the little pieces, and then we say, look in that one right there. And then when you look in that one, you can find the antibody signal or the virus signal. So this was sort of another one of these, what? So this is R11. So I do believe it's this one right here. And so you can see that in addition to there being antibody on the surface, there's antibody below the surface. What is going on here? And if you look closely, these look like dendritic cells. But it turns out to be some classic mucosal, at least mouse immunology, these manifold cells. So manifold cells, I love it when you find these things and you can just find books about it. So manifold cells grab immune complexes, especially with IgA. They pass them through to these dendritic cells, which then run to the draining lymph nodes. So we can look in the draining lymph nodes. And sure enough, we can see the IgA in those draining lymph nodes where the copper tells us to look. And then we are doing the same thing with the virus. And to find virus in tissue, we have this nice method of photoactivatable GFP, which allows us to see, you see this background in this explant, like a green dot here. This is not virus. You photoactivate, the signal emerges. That signal was not there. When you put them over each other, that is a viral particle. This is background. So it's very important to validate things. The virus is very small. Tissue is very autofluorescent. You have to validate. So now if we copper label the virus and we look in our spots, we can see immune complexes. It's kind of, I know they've been good lights, but you can see all the little red dots. These are viral particles associated with IgA, so they're being captured in immune complexes. And then what's pretty amazing is that we can go into these draining lymph nodes and we can see the IgA virus complexes within the draining lymph nodes with our photoactivation method. So we would have never looked. In fact, when the first experiment with this came up, I came in to sort of be part of the experiment, but then I had to get back to Chicago. And I'm texting with my postdoc, and she's saying, I think there's signals in lymph nodes. And I just said, start collecting them, collect them. And she said the people working there helping her were laughing. Why? We put this in the rectum. Why are you looking 2 hours later at draining lymph nodes? But we find these things in draining lymph nodes. So thank you for your attention. And I have some people in my lab who like to play with images. So here's one of our early PET scans. Thank you, sir. And so I'll go ahead and stop. And these are the collaborators that have really made all this happen. And I was in Nairobi in January, and I made a deal with this giraffe that if he would take a selfie with me, I would show his picture around the world. So there he is. Thank you. And I only went 2 minutes over, so I did okay. And I'd be glad to try to answer any questions. Thank you.
Thank you so much for beautiful presentation and these fabulous pictures. So, for antibody, like when antibody go inside the cells, how it get protected from the enzymes, like from the lysosome? And following this question, is it the antibody will cross the blood-brain barrier, like?
So I'll answer the second question first, and the answer is definitely it can cross the blood-brain barrier. It's less efficient than some other sites, but I had this debate with Dr. DeRozier, and if you go into the literature, there's antibodies there. It's just about 10% of what you would see in other tissues. And I didn't have time to show this, but there's antibodies accumulating in these very spider-like cells in the brain that are probably astrocytes or glial cells. We're in the process of sorting that out. How it's getting there, I don't know, but within 24 hours, those cells are heavily loaded with antibody. I thought they might be migrating in, but now I think they may have a projection in a capillary, and they're just sort of drawing the antibodies out. But that's a testable hypothesis, and we can look at that. As I showed you for FcRn, so some things that get taken up by the cell get destroyed in the lysosome, but other things like transferrin receptor and transferrin. Transferrin is a very famous example. It gets recycled back out. Or CD4 gets internalized, and then it goes back out. Everything on the cell surface is coming in on a regular basis, and then some things are trafficked to go back out, and some are trafficked to be destroyed, and then the cell has the ability to say, sorry, my friend, but this time you get destroyed. So sometimes it recycles, and sometimes they get degraded.
Last question, please.
So Tom, can you visualize cells moving around in vivo or in a limb using any of your imaging approaches by labeling the cells? Can you watch cells move through lymphatics and into lymph nodes?
Yes. So that method is multiphoton imaging, and there's a lot. I'm sure you've seen, the T cells moving around in lymph nodes in mice. We have that instrument, and we have that capability. You can look a millimeter or two into tissue, and in a mouse lymph node, you can see a lot. In a monkey, it's hard to get them to the microscope, and harder to see those things. But we can look at the dynamics under certain circumstances, and maybe we start to push things like the PET and other stuff. We'll be able to get to see some details like that. So, all right, thank you very much.
Okay. Thank you.